Now students, in today's lecture, we are going to discuss clinical significances of the enzyme in diagnosis of certain diseases. So, plasma enzyme level in disease state usually increases. So, whenever there is tissue damage, there will be release of intracellular enzymes into the plasma. And now this level can be used in the diagnosis of diseases. So, the level of these enzymes can also be used to estimate to determine the extent of tissue damage as well. So, we can conclude that plasma enzymes are a diagnostic tool. So, some enzymes have high activity in only one or few tissues. For example, when we talk about the liver, our liver contains an enzyme that is alanine aminotransferase, that is ALT. So, whenever there will be a tissue damage, whenever there will be liver damage, whenever there will be damage to the hepatic tissues, there will be increased content of this enzyme, there will be increased level of this ALT in the plasma. So, our heart contains different isoenzymes. Now, what are isoenzymes? Isoenzymes, also known as isozymes, these are actually the enzymes that catalyze the same reaction. No doubt they do not contain the same physical properties because they have genetically difference in the amino acid sequence. Now, we know that our enzymes are composed of amino acids. Our enzymes are composed of proteins. We have a machine, that is electrophoresis machine, that has ability to separate these enzymes because of the movement of the charged particles in the electric field. So on this property, on this electric property, we can separate the enzymes. So different organs in our body contain different isoenzymes, which I will discuss in this lecture. So for example, our plasma level of creatinine kinase, that is represented by CK, increases in case of myocardial infarction. We abbreviate myocardial infarction by MI. So these enzymes together with the other test, for example, with the ECG, that is electrocardiogram, can be used for the interpretation of different results, can be used for the diagnosis of the myocardial infarction. Here this figure is from the lipid core biochemistry. It is representing that we have a machine, that is electrophoresis machine, and then we have different isoforms of creatinine kinase, isoform 1, 2 and 3. So there will be movement of these isoforms towards the electrode. We know that these isoforms are basically negatively charged and they will move towards the positively charged electrode. In case of normal heart activity, we have a normal baseline value of these isoenzymes, while in case of myocardial infarction, there will be increased level of these isoenzymes. So, what are these isoenzymes? These isoenzymes are basically the quaternary structure. Our body contains three common types of the creatinine kinases, three isoforms of creatinine kinases, that is creatinine kinase 1, 2 and 3. CK1 contains BB polypeptides, CK2 contains 2 polypeptides M and B, CK3 contains 2 polypeptides M and N. So from this we can conclude that creatinine kinases are basically dimers. They are composed of 2 polypeptides or 2 subunits. As we can see, in CK1 we have 2 subunits BB, in CK2 we have 2 subunits MB, in case of CK3 we have subunit MM. So our brain contains the isoform BB, that is our brain contains the CK1, while our skeletal muscles contain the isoform CK3, that are MM, while when talking about the heart, our cardiac muscles contains 1/3 of the MB, that is CK2, and the rest is MM, that is CK3. Now, what happens in case of the myocardial infarction? We know that the level of proteins act as a biomarker for the diagnosis of myocardial infarction. Normally, our myocardial muscles contain greater than 5% of the total creatine kinase. So when we talk about this CK2, that are FB isoenzyme. So what happens in case of myocardial infarction? In case of acute condition, there will be increased level of this CK2 in plasma within 4 to 8 hours. So along with the feature that is the chest pain. Now, this will reach to peak activity within 24 hours and will return to baseline after 48 to 72 hours. Here, in the diagram of the level cord, this is actually the baseline value. So, we can see that in case of myocardial infarction, there will be increased level of the creatine kinase CK2 along with the certain features of myocardial infarction, that is the chest pain. So, the level of these isoenzyme together with the certain clinical features, for example, chest pain, plus along with certain other tests like ECG, can be used as a gold standard for diagnosis for the determination of myocardial infarction. We know that we have certain troponins in our body, and particularly when we talk about the heart, we have cardiac specific isoform of these troponin, that is CTN. They are released in the plasma in case of the cardiac damage. So this CTN level increases or appears in plasma within 4 to 6 hours after MI, after myocardial infarction, and will reach in peak after 24 to 36 hours. Now, and this amount will be remain elevated in the plasma for 3 to 10 days. Now, so we can conclude that this elevated CTN level in combination with clinical presentations plus ECG can be used as a gold standard for the diagnosis of myocardial infarction. So from the whole discussion, we can conclude that the level of these enzymes, the level of these isoenzymes can be used for the diagnosis of certain diseases.